Hello everyone, today I want to introduce you to a fascinating community science project called Spyfish Aotearoa, which is an important initiative aimed at understanding and conserving marine biodiversity in New Zealand's marine reserves. In the following, I will share my experiences and the project's impact on marine conservation. To begin with, Spyfish Aotearoa is a citizen science project hosted on the Universe platform. It's aimed to monitor marine biodiversity in New Zealand by collecting data on fish species observed in marine reserves. Volunteers like myself are tasked with watching short video clips captured by underwater cameras and identifying various fish species within these clips. The main goal is to gather data that can inform conservation strategies and ensure the health of marine ecosystems. This hands-on project allows volunteers to contribute directly to important scientific research by providing data that might otherwise be challenging for a limited number of researchers to collect on their own. Now, let's delve into the scientific context that makes this project so important. The urgency of, of the Spyfish Aotearoa project arises from the significant decline in marine biodiversity due to factors like overfishing, pollution, and climate change. Overfishing, for instance, has led to a reduction in fishing population and threatens the balance of marine ecosystems. In New Zealand, where the project is based, marine reserves are important for conserving biodiversity and protecting vulnerable species. According to Aotearoa, marine reserves that are well enforced, large, and isolated from fishing pressure are more effective in promoting biodiversity and increasing fish populations. An example of this is the Goat Island Marine Reserve, where studies have shown a noticeable increase in the number and size of snapper and other fish species since the reserve's establishment. Spyfish Aotearoa aims to extend the successful to other marine reserves by providing the data needed to evaluate their effectiveness and guide conservation policies. By analyzing the footage and identifying species, we can determine how well these reserves are functioning and identify areas that require more protection or different management strategies. Translating to my personal experience, volunteering with Spyfish Aotearoa has been an enriched experience which enhanced my understanding of marine life and the challenges of biodiversity conservation. Over several weeks, I spent approximately 9 hours reviewing and analyzing over 140 video clips to identify various fish species. Before participating in this project, I could not tell the difference between the fish. Through dedicated observation of video clips, I learned to recognize these fish by their body shapes, size, and movement patterns. For instance, at first, I found it difficult to differentiate between the smooth leather jacket and the blue coat. Both species have distinctive features, but they can be easily confused at first glance. The smooth leather jacket has a sleek body with a retractable horn-like dorsal spine and varying coloration, whereas the blue coat has a blue-gray back with turquoise band behind its head in its mature face. As I became more familiar with these differences, I learned to spot them quickly. This skill in identifying fish species has been crucial for contributing accurate data, which is essential for effective conservation planning. Additionally, analyzing video footage has given me insight into non-invasive data collection methods in marine research. Unlike traditional diving surveys, Bited underwater camera captured nature fish behavior without human disturbance. These efficient methods offer unique perspective about attracting various species and revealing intersections that aid scientists in understanding ecosystem dynamics. Moving forward, the data gathered from Spyfish Aotearoa is being used in several impactful ways. Firstly, it helps evaluate the effectiveness of New Zealand's marine reserves by comparing the abundance and diversity of fish species within protected areas to those in fished areas. This comparison can reveal whether marine reserves are successfully protecting and enhancing fish population. For example, if a marine reserve shows a higher number of juvenile fish compared to surrounding waters, it might indicate successfully breeding and a healthy environment. Furthermore, the data has application in policy making, where insights can inform the creation of new marine reserves or the expansion of existing ones. Scientists can also use the information to study the impact of environment changes, such as rising sea temperature on fish species distributions. This aspect of the project is critical for predicting how climate change might affect marine life and planning conservation strategies accordingly. Reflecting on my experience with fishing Spyfish Aotearoa, I am grateful for the opportunity to contribute to such an impact project. One significant lesson I learned is the potential of community science to drive positive change. By engaging the public in scientific research, projects like Spyfish Aotearoa foster a sense of responsibility and connection to the ocean. This engagement is crucial for building support for conservation initiatives and promoting sustainable practices. I also realized the immense value of collective efforts in addressing complex environmental issues. The project taught me that individual contribution, no matter how small, can significantly impact scientific research and conservation. Moreover, my experience with Spyfish Aotearoa reinforced my appreciation for marine ecosystems and the role of citizen science in advancing scientific knowledge. It has inspired me to continue participating in community science projects and contribute to preservation of our planet's biodiversity. Without a doubt, I would participate in a community science project again. The experience with Spyfish Aotearoa has been fulfilling and impactful. In the future, I am particularly interested in projects such as a penguin watch and a frog find, which focus on wildlife conservation and biodiversity pres- preservation. These projects align with my interests in wildlife conservation and offer the opportunity to contribute to vital ecological research globally. In conclusion, by getting involved and supporting projects like this, we can all make a big difference in preserving our oceans for future generations. Thank you for your attention.